All right, I'm going to talk about the cellular cycle. Let me get off of this. Yesterday, you read about and watched about the cell cycle, but I'm just going to go over and make sure you really are comprehending what you're reading. At any time during the video, obviously pause, take notes. I'm going to use the graphic from the reading, so you can either copy and paste that over or just grab it or draw it, whatever you like to do, but you have to take these notes. So the cell cycle, first of all, is 24 hours to days, depending on what type of cell it is. And then the majority of the cell cycle, you can see here, is going to be the interphase, okay? And that's going to be G1, S, G2. So right here, I wrote down that most of the cycle will be spent in interphase. The cell grows and copies its DNA. The cell grows to normal life. The cell is going to do whatever it needs to do to be normal. S is DNA synthesis, where the DNA will make another copy of itself. Why would a cell need 2 copies of DNA? Well, it would need that because it's going to split into 2 cells. So each cell needs its own copy, so it's going to split that way. G2 is more growth or preparation for mitosis, right? And that's all interphase. So when you think about all the cells in your body, the majority of cells are going to be doing interphase. After interphase is mitosis, and that was listed as M in your reading. Mitosis is where you're making more cells, and they're exact copies. So mitosis needs to be exact copies of each other. And the cell divides into DNA and cytoplasm, forming 2 new cells. That's called mitosis. That was the second video link. So that's going to be prophase, metaphase, anaphase, telophase. I'm going to talk about mitosis later in the video. So if you look over here at the graphic, I put 3 X's for different checkpoints. In your reading, the checkpoint was controlled by some different molecules, and I will explain those. So if I scroll down, there are checkpoints. The checkpoints check if DNA is in the correct stage. The whole cell cycle is designed to be around the DNA. DNA is always going to be the star. So at any checkpoint, think about if you've ever been in the airport and you were at security check, you're going to need 2 things. You're going to need things to get through that checkpoint. The cell cycle is the same way. If the DNA is good, you're going to get a yes. Okay, you are going to get through the checkpoint, but you're going to get through the checkpoint with this over here. And I will highlight that. So all of this stuff over here, that is about what it takes to get through the checkpoint. So you're going to have CDK and cyclin, and this is called the lock and key method. CDK is the lock, cyclin is the key. So if you think of the classroom and think about the door, the lock is going to always be there. And the same thing with CDK. The cell always has CDK. But the key to my room, or key to any door, will probably move. It will move with the person who has the key. And the same thing with cyclin. Cyclin will be there if the checkpoint is met, or you can go through the checkpoint. And that's why it's called the lock and key. CDK and cyclin together means it can go through the checkpoint. So just like at the airport, you need a boarding pass and you need an ID or a passport. You need 2 things to get through. Now, if you look up here, there are 3 checkpoints. So obviously there's 3 different doors. When you think about what would a door need, there's probably going to be different locks and different keys. The same thing with the cell. There's not just one type of cyclin. There's not one type of CDK. There's multiple. Now, what if the DNA is bad, or the DNA is wrong, or the DNA is broken? Then you're getting stopped at the checkpoint. You're not going through. You're going to get stopped. Okay? So no DNA is bad. Okay? No, the DNA is bad. The DNA is broken. The DNA is wrong. There's going to be some options. Number one, the cell will repair the DNA. So if the cell has the DNA not where it's supposed to be, maybe it's not lined up, maybe they're not done with making a full another option for it, the cell will repair its DNA. Just like at the airport, if you don't have your ID, you're looking around in your luggage and you find your ID. And the cell's like, great, go on through the checkpoint. If the cell has tried that and it can't, the cell will experience something called apoptosis, meaning the cell will explode. The cell will not be able to fix itself. The cell has no control. So what it's going to do is do programmed cell death. And I like to think a pop, like the cell pops, which is good. You want a cell that can't be controlled to die. Third, the cell goes unchecked. It reproduces. The reproduction of the cell is uncontrolled. And that's when we have a cancer cell. Okay? So a cell that has broken DNA no longer is affected by checkpoints and goes through the cycle completely unchecked. That cell is broken, but we're going to rename that. That becomes a cancerous cell, a cell that no longer follows the rules of the cell cycle. Now, that can happen as the DNA can be broken accidentally. It can be random, but there are things that humans do to increase our rates. The top 3 cancer-causing activities, or whatever you'd like to say, that we do is number 1, tobacco. Tobacco breaks DNA. 2, obesity. Long-term obesity. So we're not saying about weight fluctuating. We're saying long-term obesity will increase the rate of broken DNA. And then 3rd, alcohol. Drinking alcohol will break DNA. So these 3 things are the top causes of broken DNA, and hence why they're considered the top 3 causes of cancer. There's other things, right? Radiation, but who's walking around getting uncontrolled radiation? Okay, and there's other chemicals. So in the future, you want to make sure you're working safely at a job if it's involved different chemicals. But overall, for most people, these are the things that are going to cause broken DNA. So that was a quick summary of the cell cycle. And again, go back, pause it, take notes. Now I'm going to go through mitosis. I'm going to quickly go through the phases of mitosis. This is the second video you watched. It's going to be prophase, metaphase, anaphase, telophase. It's going to be right here. Okay, so again, it's going to be right up here. Okay, I'm going to scroll down. First of all, I like to think mitosis PMAT. I like to think of a puppy getting potty trained, so like a puppy peeing on a mat, or a toddler getting potty trained, a toddler peeing on a mat. If you can remember PMAT, you remember the names of the phases in their order. Okay, so that's mitosis. Mitosis is going from one cell and making 2 identical cells. So first, prophase. It's the breakdown of the nuclear envelope, and that's going to be in yellow, and it's going to be yellow in the picture. The DNA condenses. The centrioles start releasing spindle fibers, so that looks like that, and the centrioles are there. You should remember that from your prior knowledge. Okay, so your prior knowledge from last semester about cell organelles, hopefully. If not, that's okay. The DNA condenses into chromosomes. DNA can have different labels. If you remember, chromatin is loose DNA. And so when DNA condenses, it condenses into what's called chromosomes. So prophase is right here, okay? Your DNA is condensing. The centrioles are going to start releasing. The nuclear envelope breaks down. Okay, then I'm going to go over to metaphase. Metaphase, think M, middle. So metaphase, middle. The chromosomes line up in the middle, and that's where I have that in blue. The spindle fibers are going to connect the chromosomes, and that's why I have that in pink. I'd like to think about the centrioles as being the fishing reel, the spindle fibers as being the fishing line, and then the chromosomes like being the fish, okay? And that you're lining the chromosomes up. They're all on the line, the spindle fibers. So that's why when you see this in the picture, you're going to think, okay, all the chromosomes lined up in the middle. That's metaphase. Anaphase is away. The spindle fibers pull chromosomes apart into opposite sides of the cell. So you can see here that the spindle fiber is shortened and they got pulled. And you can see that this cell is getting longer. And now you have, each of these is going to have what's called a half of the chromosome. That's okay. They're called sisters because they're identical. So you have identical sisters. They're going to each cell. So each cell has the identical DNA. So that's anaphase. And then the last one is telophase. Telophase is the opposite of prophase. So the nuclear envelope is going to rebuild. Chromosomes will loosen into the chromatin. Spindle fibers will break down. And then you're going to start to get a furrow. It's going to start to want to split. It's not fully split, but it's going to start to split. So that's PMAT, the process of mitosis. When it's fully completed, when it's done, it's called cytokinesis. When the cells are fully split, including the cytoplasm and including everything else, it is now going to go back into interphase. So again, cytokinesis is when the cells are fully split, including the cytoplasm. Okay? And cyto means cell. So when you're thinking about what order it happens, it's interphase, then mitosis, and then cytokinesis, and then interphase again, mitosis, cytokinesis, and it just happens over and over. The key thing is that the cells that are produced are identical. So that's really important. Cells that are split, that you have two of, are identical. And this process is going to go over and over and over again. Okay. Make sure you've taken good notes on this. Okay. And this is a summary of cell cycle and mitosis.